Hello everybody, welcome to Life Sciences. My name is Kathy Hasty and today we are spending the next hour doing the nervous system and the endocrine system. Before we start though, a huge shout out to Liberty. They're our major funders and it's because of them that all of this is possible. Please also a reminder to download the Tenfold Education app. It's got a tutor function, so you can send us questions via the tutor function. It's got lessons for life sciences and for maths and for physics. So there you go. I mean, it should be, it's like your tutor in the pocket. And then WhatsApp. You can WhatsApp us your questions. Please take a good photo of your questions on a flat surface. Make sure it's clear and send them through to us so that we can use them here. And then, of course, we have YouTube. And all you do is you go at Mindset Learn, and we have all the lessons that we do here during the week. They uploaded and they cover pretty much every topic you can think of within the syllabus, of course. So there you go. And you know what? I'd like to hear from you via our Facebook page, which I check. And it's always nice to get to know you. So just let us know who our learners are, the maths learners, the accounting learners, the English, the life science learners. Those are the learners I'm concerned about. So just message us and just say, how's it? I liked the lesson. I didn't like the lesson. Please, can you do X, Y, Z? So it's just good to get to know you because you're on that side of the camera. All right, so let's look at our first question today. It's on the nervous system. Oh, wait, you know what? I have forgotten my motivation for today, a motivational quote, and here it is. If you're not willing to learn, no one can help you. But if you are determined to learn, no one can stop you. And then the future is yours. It's like the oyster. You can do whatever you want to. So people, focus now. This is your last year so that you can do well. We want you to do well. In spite of COVID, in spite of all the nonsense, we want you to ace your exam. So here we go. First question. I have the brain and we have, where are my glasses? You see this is old age. You've got the diagram below represents two possible pathways, A and B, which a nerve impulse may follow in the human body. Okay, so let's have a look here. So here's our diagram. So what can you see? Before we even look at the questions, what can you see? So here's the brain, and we're looking at this part of the brain, which is the cerebrum. Okay, and we've got pathway A. Well, clearly this goes from the receptor, which converts the stimulus to an impulse. That then goes along, and it goes along this sensory neuron through the dorsal root ganglion. That's the sensory neuron. And it goes through a connector neuron, and then out along to, which is the motor neuron, to the effector. And the effector can be a gland or it can be a muscle. Let's just move the screen up a little bit so you can see. Okay, now, this pathway A, because it only goes through the spinal cord, is going to be a reflex arc. Now remember, the reflex arc is the path, and a reflex action is going to be the result, which it will be caused by that reflex arc. Then pathway B, we have our receptor, and we have our sensory neuron passes through the dorsal root ganglion, and then it goes from here via a sensory neuron. Remember, sensory neurons will always transmit impulses to the spinal cord and to the brain. So sensory neuron, and it goes all the way to the brain. When it gets to the cerebrum, the message is interpreted, and we have connector neurons here, which they're not showing, and then it travels along the motor neuron, down through the spinal cord. One is the spinal cord. Okay, down, until it gets here, and then it goes along, still the motor neuron, and it goes to the effector. And this effector, this is going to be for a movement. So it's going to cause the muscle to move. Let me just write a little bit. See, when I write too fast, the pen skips. Movement. Okay? And this pathway here is going to cause a voluntary movement. So this is just, I've done it again, mint. Okay, so these are two pathways. One is just a normal pathway to cause a voluntary movement, like I want to lift my arm or something. And this pathway A is a reflex arc because it only uses the spinal cord. Okay, so here's your spinal cord here. Okay, now let's check our questions out. Which pathway, A or B, represents a reflex arc? We've already said it is pathway A. Okay, done and dusted. Give a visible reason in the diagram for your answer. Well, it only goes to the spinal cord. So it's very, very simple to say impulse travels from receptor to the effector via the spinal cord only. And this is the important word you had to put in, because your reflex arc resulting in a reflex action is only, the messages, the impulses only travel via the spinal cord. That's it. That will cause a reflex action. So reflex arc is the pathway, reflex action is the result. Okay, and remember your reflex action is a very quick, rapid response. So describe the there we go. Describe the importance of a reflex action to the human body. Well, that's easy, and you must know this off by heart. This is a definition. You say a reflex action is a rapid, automatic, in other words, you don't think about it, and involuntary. Those are your three important words. It's rapid, it's very quick. It's automatic, there's no thinking. It is involuntary, which again reinforces the fact that it is no thinking. Response to an external stimulus. Okay, to, and this is also important, to prevent injury and protect the body. So there's no thinking about it. It's a rapid, automatic, involuntary response to an external stimulus. So in other words, if I shine a bright light in your eye, what's going to happen? Your pupil is going to constrict. Why? Because of the pupillary mechanism. And what's that going to do? Well, the circular muscles will contract, the radial muscles will relax, and the pupil will constrict. End of story. It lets in less light. If you stand on a nail or a thorn, or you put your hand on a hot plate, all of it. You're going to pull your hand away long before you even feel any kind of pain. So it's a rapid, automatic, involuntary response to an external stimulus to prevent injury to your body. It's as simple as that. So you're protecting your body. Okay, now, identify the part of the nervous system represented by 1. So let's just quickly check. Oh, 1 is the spinal cord. This thing here, spinal cord. And remember, your central nervous system consists of the spinal cord and the brain. So this is an easy peasy mark. All right, now, explain one way in which the myelin sheath is important for the functioning of neurons. Now remember, on the neuron, you have the axon. Okay, you have your terminal end plate this side. Okay, and here if it's a multipolar or unipolar, doesn't matter. But then you've got, somewhere you're going to have your cell body here somewhere. Okay, so this is the axon. And when your axon, around the axon, you have the Schwann cells which make up the myelin sheath. So there's your myelin sheath. Okay, so axon, let me just extend the line to there. Okay, so myelin sheath. So what does it do? It insulates because it wraps around the axon. So if this is the axon, it wraps around the axon, so it insulates the axon. That's very important. And it provides an electrical insulation so that those impulses don't sort of go sideways. They stay within the axon, traveling either to the central nervous system via the sensory neurons or away from the central nervous system via the motor neurons. So the first one that you need to remember, or it's easy to remember, is it acts as an electrical insulator. It insulates, it keeps it all nice and cozy. The second thing it does, and I'm going to give you 3, it allows impulses to transmit. Because it is an insulator, an electrical insulator, it allows impulses to transmit quickly and efficiently along the nerve cell. Okay, so it provides this insulation, and that's what allows it to transmit quickly and efficiently along the nerve cell. And lastly, the myelin sheath is responsible for repairing damage to the axon. Now, your normal axons of your sensory and motor and connected, or your sensory and motor neurons are going to have a myelin sheath. But in the brain and the spinal cord, they are unmyelinated. And that is why when there is damage in the brain or there is damage in the spinal cord, it cannot be repaired. Okay, it's because of the myelin sheath. Right, or the absence of the myelin sheath. Okay, and now, okay, here we go. Describe how a person would be affected if the axon on neuron 2 was cut. So let's go back here. Yeah, okay, here. So there's 2, so it's a motor neuron. So if we cut the motor neuron, what's going to happen? You're going to have your receptors going to convert the stimulus. I'm going to use another color. It's going to convert the stimulus into an impulse. The impulse is going to travel along the sensory neuron to the connector neuron and then to the motor neuron. And well, it can't go any further because it's been cut. So what's going to happen here if it's cut? We go here, cut. So if it's cut, what do we have? Well, the impulse can't get to the effector. And if it can't get to the effector, there's going to be no reflex action. So let's put that in exam speak so we can get some marks. We're going to say the stimulus would be converted to an impulse and move along the sensory neuron and connector neuron but not to the effector. Therefore, it will result in damage. And why? Because what is the purpose of a reflex action? If you cut the motor neuron, then there is no way to get that impulse to the effector. And remember, the effector has to undergo a reflex action to remove the limb from the painful stimulus. So if the receptor's working and the sensory neuron's working and the connector neuron's working, but the motor neuron isn't, there is no means to cause the effector to respond. Okay, and well, since a reflex action is about damage, that's the problem. Okay, now it says here, describe pathway B. Now, if we look at pathway B, what do we have? We have the receptor, and it travels, a receptor converts the stimulus to an impulse, sensory neuron, spinal cord, all the way up, cerebrum, and then motor neuron and effector. So now we put that into words. It's exactly what you see in the diagram. And that is going to be as follows. You're going to say receptors receive the stimulus. Okay, and what do? Point 2, and convert it to an impulse. So now what? Impulse is transmitted along sensory neurons via the spinal cord to where? To the brain. Or you can say cerebrum. Okay, so now it gets to the brain. Great. The brain interprets the impulse message. Message, okay, and sends an impulse along the motor neuron just move this up a little bit, along the motor neuron to via the spinal cord to the effector to bring about a response. And there you go. So the receptors receive the stimulus, they convert it to an impulse. Then the impulse is transmitted along sensory neurons via the spinal cord, which is the central nervous system, to the brain, or you can say cerebrum. Both are correct. The brain then interprets the message, okay, and sends an impulse, because that's the language, it's the pathway, the impulse along the motor neurons via the spinal cord to the effector, where we will now have a response. Please remember the following, guys and girls. Here's your brain and your spinal cord. Anything that carries messages to the spinal cord and to the brain, okay, those are going to be your sensory neurons. Okay, then in the spinal cord and in the brain, you have connector neurons. Okay, and you can also call them interneurons. And think of these as on and off ramps. So onto a highway, on and off ramps. So it's the link between the sensory neuron and the motor neuron. And then we go with, let's go with orange. And coming back from the brain and the spinal cord, you have your motor neurons. Now it's easy to remember. Sensory is your senses. Motor, think of motor as move. And your motor neurons will cause a movement because they will always end in an effector. Where your sensory neurons are always the receptor receives the stimulus, so receptor for receive, receives the stimulus and converts it to an impulse. And motor neurons and the effector only always work with impulses as well. And if you remember that, you actually cannot go wrong with any question regarding the nervous system. All right, now, how are we going for time? I think let's do one more question and then we can go to time. We can go for a break. Okay, so this question was sent, the other question was sent by Tito, by the way. This question was sent in by Dudu. So we've got a chap and we've got a gorgeous girl who sent us a question. Unlike last week when we only had boys that sent us in questions. So here we go. Study the following diagram representing glands in the human body. So this one here sits around the trachea, so it sits here like this, like a little bow tie. So if you were going to wear a bow tie, this is where your thyroid is. Okay, this one sits above the kidneys, and kidneys are called renals. Everything to do with the kidneys, renal. So we've added them to the renals, so this is your adrenal gland. And C, well, you don't learn about the liver and the gallbladder and the small intestine in matric, but grade 12, but what you do learn about is the pancreas. And what makes the pancreas so special is it is the only gland in your body that is both an exocrine and an endocrine gland. It's an exocrine gland because it has ducts, and pancreatic juice, which are digestive enzymes, are released out via the ducts, the pancreatic duct, into the common bile pancreatic duct and into the duodenum to neutralize acid from the stomach. But the endocrine part of the pancreas has got the alpha and beta cells of the islets of Langerhans, and they are responsible for controlling your blood sugar levels. So, or your blood glucose levels. You can call it both. Glucose, sugar, same thing. So it's identified the glands. Well, we had the thyroid gland. And then we had the adrenal gland. And I wonder what hormone the adrenal gland releases. It is adrenaline and aldosterone. So both A's, you actually can't mistake that. And then C was the pancreas. Our very special pancreas, because it is both an exocrine and an endocrine gland. It's very, very special. All right, now, the functioning of the gland in diagram A is controlled by another hormone. So we're talking about the thyroid. So I'm going to just put thyroid here, just in case you forget. Okay, the thyroid. Provide the name of this hormone that controls the thyroid. Well, it is the thyroid stimulating hormone. So thyroid stimulating hormone, you can just simply write TSH. Thyroid stimulating hormone stimulates the thyroid to produce thyroxine. And what does thyroxine do? Thyroxine increases your metabolism. All right, so which gland is responsible for the secretion of the hormone identified in question 2.3a, which is thyroid stimulating hormone? Well, that's an easy one. It is the pituitary gland. And the pituitary gland is also special because it is called the master gland. Why? Because it releases so many hormones that stimulate other organs to perform their functions. Okay, so that's why it's the master gland. And what controls the pituitary gland is the hypothalamus. And the hypothalamus controls your life. It controls every single thing about you, is the hypothalamus. All right, so here we go. What condition activates the secretion of adrenaline by the gland in diagram B? So diagram B was the adrenal gland. And what's going to stimulate the adrenal gland to release adrenaline? Well, guess what? It is the fight or flight. So you box or you run. Your fight or flight hormone is released when a person is in danger. So you hear a snake hissing behind you, okay, or in a stressful situation. So think about it. I mean, if you were to have a car accident, heaven forbid, but if you were in a car accident, you have a huge amount of adrenaline that's released. And you're like all over the show, and you have a whole lot of energy, and your blood sugar's high, everything's high. And then when people have had a really big fright, they go pale, which means all the blood is directed to increase the blood pressure in your muscles, and you get nauseous. So that's because the blood supply to the digestive system in the stomach, the blood vessels contract to redirect the blood to your muscles so you can fight or flight, fight or run, okay? And that's normally why, you know, in movies when you see people, when they get a huge fright, they vomit, okay, afterwards. Well, that's because of the adrenaline. Your stomach, everything just contracts as those blood vessels pull away and constrict. Everything contracts and then you just heave. But the paleness and that cold sweat, all of that is a response to adrenaline. All right, so in case they ask you for a situation, but it's whenever you're in a stressful situation or a dangerous situation. And then, how does the secretion of adrenaline affect the heart rate and the breathing? Well, it's not just the breathing, it's the breathing rate and depth. Well, guess what? Both increase. Why? Because the heart needs to beat faster to get the glucose and the energy and the oxygen to all the parts of the body. And your breathing rate and depth increases because you need more oxygen, because your muscles need to work, especially if you've got to run and get out of the situation. So it's your heart rate and your breathing rate and depth. Okay, not just breathing. If you've got to answer that in an exam, it would be, if they said to you, how does the secretion, what is increased by the secretion of adrenaline? You'd say heartbeat rate increases, breathing rate and depth increases, okay? That's your perfect question. Now, let's quickly get through this. We're heading towards an ad break soon. Okay, Luke, a grade 12 learner, did not eat breakfast. Bad, 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 bad. Never, ever, 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 ever don't have breakfast. You can not have supper, but you must have breakfast. So, explain how his blood sugar will return to a normal level. Now, if he hasn't eaten breakfast, his blood sugar is going to be low. So, here are your pointers. When the blood glucose level is low, the alpha cells of the islets of Langerhans, these are your specialized endocrine cells in the pancreas, are stimulated to secrete. Now, people, I'm going to write it in capital letters. Glucagon. Okay? So, look at the word. Glucagon. When glucose is gone, that's how you remember it. So, when's glucose going to be gone? When your blood sugar level's low. Or when glucose is gone, your blood sugar will be low, or your blood sugar is low because the glucose is gone. So, when blood sugar, because I know a lot of learners confuse the functioning of insulin and the functioning of glucagon. Okay, so when glucose is gone, your blood sugar level's low. You want to bring it back to normal. The islets of Langerhans, the alpha cells in the pancreas, will be stimulated to release glucagon. Okay, and what does glucagon do? So hang on, glucagon into the blood. Okay, let's put a full stop here. Glucagon, now remember, all hormones always travel in the blood to a target organ. So, glucagon is transported in the bloodstream to the target organs, the liver and muscle cells. Okay? And what does it do? Where it stimulates. Now remember, hormones can't do anything. They stimulate or inhibit. Stimulate the conversion of stored glycogen. Because remember, when glucose is stored, it's stored as glycogen. Stored glycogen, why? Because it's osmotically inactive. Stored glycogen to glucose. And what happens then? Well, this increasing the blood glucose level to return it to normal. And this, my friends, is what we call negative feedback. Okay, so when the blood glucose level is low, the alpha cells of the islets of Langerhans of the pancreas release glucagon. into the blood. Why? Because it's an endocrine gland. So into the blood where glucagon is transported in the bloodstream, because it can only travel in the blood, to target organs. It's got lots. It's got all the muscles in the body, and it's got the liver cells, where it stimulates the target organs to convert stored glycogen to glucose, okay? And what happens then? Well, you've got all this glucose now in the blood, so the glucose level increases, and you don't feel like you're going to fall down and fall asleep. Okay, next question. Differentiate between an exocrine and an endocrine gland. Okay, first of all, your exocrine gland, now you could do a table. Oh man, what table? Your exocrine glands, first of all, they have a duct, okay, that's important, through which useful substances are secreted to where they are required. And example, sweat glands, salivary glands, tear glands, okay, all those glands that secrete useful substances, all your enzymes in your body are secreted by exocrine glands, okay? And then endocrine glands, people, you must know this, are ductless and secrete not useful substances, they secrete hormones directly into the blood to be transported to target organs. And examples, we had, what did we have? We had the thyroid gland, adrenals, pancreas. Okay, so there you go. Simple, simple, easy peasy, there we go. How many marks is that? I think 17 marks in the bag. Thank you for coming. All right, peeps, we're going to go for a quick ad break, and I'll see you all in a few minutes.
Did you know that Lesotho is the only country on Earth that is completely over 1,000 meters in elevation? The mountainous southern African nation of Lesotho is one of the 3 countries in the world that are enclaves, independent countries that are entirely surrounded by another country. It is the only country in the world that lies completely above 1,000 meters. Its lowest point is at 1,400 meters.
No one ever asks why we do the weird things we do. Like, why once is never enough? Or doing 2 trips is just one too many. Why do we, before we use these, or say 5, when we mean 20? Why do we have second thoughts? And think that lift buttons make them go faster? Why don't we walk there, cross our fingers, touch wood, and struggle to hide our true feelings? At Liberty, we want to know all the weird and wonderful things that make us human. Because human beings, being human, are what we make financial solutions for. Liberty. In it with you.
Welcome back, folks. Before we start, a shout out to Liberty, in it with you, as always, and they have been for many, many years. Don't forget to download your Tenfold Education app. It's got a tutor function. You can submit your questions. You can live stream. I mean, it is an awesome little app, and it's all free, also provided to you by Liberty. And then don't forget WhatsApp. You can message your questions through to us. And we've got the YouTube channel, which is at Mindset Learn. And yeah, you can get all the lessons we do, etc., etc. So, and send us a message on Mindset Facebook page as well. It'll still be nice to hear from you. Okay, fokies and focus. There we go. YouTube and at Mindset Learn. Alrighty, now we're going to carry on. This is another question, and this question is sent in by Adelaide. So the girls have outdone the boys today. We've got 66% girls and 33% boys. Come on, guys. Right, so it says here, diabetes insipidus. Now be careful. You have learnt about diabetes mellitus, which is when insulin doesn't work properly. This is diabetes insipidus, which they tell you in brackets here is the production of dilute urine. It's a disease caused by, look at this, a lack of antidiuretic hormone. Now, antidiuretic hormone is, and I'm going to write it here, and I want you to remember, please never write this in an exam, but it's a way to remember it. It is an anti-urinating or weeing hormone. Okay, it doesn't want you to urinate. It doesn't want you to make a wee. Why? It wants you to keep the water inside the body so that you can sweat. So, on a hot day, you're going to release lots of antidiuretic hormone because you don't want to lose the water to urine. You want to keep that water in your body so that you can make sweat to cool your body down. On a cold day, well, guess what? You're drinking a lot because you drink lots of coffee and tea and Milo and hot chocolate and stuff to keep you warm. So you're not going to be sweating a lot because it's cold. So you don't have to cool the body down. So what happens? You make very dilute urine, and then you're going to have hardly any antidiuretic hormone because you need to get rid of all the excess water, otherwise you're going to start swelling. Okay, so remember that antidiuretic hormone, or ADH, is an anti-urinating hormone. It wants to keep the water in your body. So the more ADH you have, the more water you keep inside. The less ADH, the more water will go out as dilute urine. So when a person has diabetes insipidus, they have a lack of antidiuretic hormone. So there's nothing to keep the water in the body secreting into the plasma. Plasma is the blood. Remember, blood is made up of plasma, and it's made up of red blood cells and white blood cells and thrombocytes. Okay, scientists wanted to determine the effect of this disease on the volume of urine that a person produces per day. Now, the investigation was carried out on 5 male participants over a period of 30 days. So 30 days tells you about the reliability, which is good. 30 days is a long time. Okay, so that's a tick for reliability. The males were a similar age and weight, so that will talk to validity. Remember, when we talk about validity, it is generally, it's got the word similar or it's got the word same. So whatever you must keep similar or same talks to how valid your experiment is. The average, and here we go, here is your dependent variable, the average daily urine production, okay, or the average volume of urine produced. Okay, there you go. That is going to be your dependent variable. And this here is your dependent variable. Okay. And then whether the person has diabetes insipidus or not, these individuals were selected by the scientists. Now it says here, normal daily urine production is between 600 milliliters and 1,800 milliliters. So you've got to just understand. Oh, by the way, when they talk about the average daily urine production, that is also going to be talking about the reliability of the experiment, which is good. You calculate the average, and then it is more accurate. So here, they're giving us in liters per day, but they tell us it's 600 to 1.8 to 1,800 mils. So 600 mils is 0.6 liters, and 1,800 milliliters is going to be 1,8 liters. So look at this. Immediately you see here's 2.9. 2.9 is way over the normal daily production. Okay, but if you look here, 1.5, 1.3, 1 and 2 are all between 0.6 and 1.8. Okay, so these guys are all okay. They're normal or within the normal limits. Okay. Okay, but this one here, this is the chap that's got diabetes insipidus. So, which individual would the scientists suspect as having diabetes insipidus? Well, clearly, individual 1. All right, because he's so high. Oh, here we go. Give a reason for your answer. Well, you can say he produced, or this individual produced above the normal range of, what was it, 600 milliliters to 1,800 milliliters per day. Okay? So, I mean, that's easy. So far we now have 2 easy marks in the bag, and we move on. What does the word plasma tell you? Oh, I told you. Plasma, you're talking about the blood. So what does it tell you about the type of gland that releases ADH? Well, the fact that ADH is a hormone, it means it is an endocrine gland that secretes ADH, or a hormone, directly into the blood, or you can say bloodstream. It's up to you. Okay, but when they talk about plasma, remember blood, we have plasma makes up about 90% of blood, and then it carries your gases and your nutrients and your wastes are all in the plasma. It's all dissolved in the plasma. And then you have your red blood cells, your white blood cells, and your thrombocytes, which are for blood clotting, and your killer T cells and whatever other cells that are floating around in your blood. But the plasma is the fluid which carries everything, made up of 90% water. Okay, then identify the dependent variable. What did we say it was? We just have to look here at the table. There we go. It's the average volume of urine produced. So, dependent variable, average volume of urine produced. Okay, Now, identify 2 factors that should be kept constant during this investigation. Now, what are we trying to do in this investigation, or what are they trying to do? They're trying to determine the average volume of urine produced per day. Okay, so what is going to lose water and change how normal the urine production is? You're going to have sweat. What's going to cause sweat? Well, heat will cause sweat. So temperature. Exercise will cause sweat. So let's look at this. What's going to cause, well, first of all, what's going to give us water? So number 1, let's say 2 factors. So I'm going to say here, volume of liquids, or you can say fluids, volume of liquids that each participant drank must be the same. Now remember, when we talk about must be kept constant, we're talking about what's going to make our make the experiment valid. So you're talking about the validity. So what makes it valid? It must have the word similar, as I said earlier, or same. So the same volume of liquids, number 1. Number 2, the same type of liquids. Doesn't help the one guy drinks beer and the other guy is drinking just plain water and the other one's drinking sweet and fuzzy cool drink. You can't have that. They must all be drinking exactly the same fluid and the same amount of those fluids. All right, because a beer, for example, contains alcohol, and alcohol is an ADH inhibitor. That's why when people drink beer, they need to go and urinate so often. Okay, or any alcohol really. So the same type of liquids, the same volume of liquids, they must be kept in the same temp in the environment. So in the room, wherever they're staying, it must be the same temperature. What else? What else is going to cause? We said it was the same amount of physical exercise. Okay, so the first 2 are about keeping the same fluids. The second 2 are about making sure that you're not using that water in the body to perspire or sweat, and you then are going to have a more concentrated urine output. Okay, so everything must be kept the same. That talks to validity. Provide 1 precaution. Oh, please, people. I'm not even going to write this down. What is the precaution? You've got to make sure that the lab assistants are wearing gloves and goggles and lab coats, because what are they working with? They're working with someone else's urine. So you must make sure that everything is sterile. You've got to make sure that the samples are collected accurately and that they are accurately labeled, so that they are correctly labeled. I mean, it doesn't help that you mix up 1 and 5's urine samples. You've got to make sure that the equipment is sterilized and hygienic. So, I mean, really, the precautions, just think of the safety measures. If you were working with someone else's urine, what would you put in place? That is a bonus mark. Okay, then state 2 ways in which the scientists improved reliability in their investigation. So if we look back at their investigation, what did they do to improve reliability? Here we go. Let me use orange. They conducted the experiment over 30 days and they calculated the average daily urine production. So there are your 2 answers there. So let's quickly put them down. Investigation conducted over 30-day period. Okay, and average daily urine production was calculated. Okay, so when they work with an average, or you're doing it over a nice period of time, or the other ways in which to ensure reliability is to repeat the experiment a couple of times, or to use more people. Oh yeah, we're running out of time. Okay, now, oh my word. Draw a pie chart of the information in the table and show your calculations. Oh. Okay, so first, calculations. You must know how to draw. They don't actually ask line graphs in the exams, but they do ask bar graphs and histograms. You've got to know the difference between a bar graph and a histogram. Okay, so that's a bar graph is continuous and a histogram is discontinuous, so like blood types, and a pie chart. And I'm not a person who's very fond of pie charts, but here we go. Okay, so what are our numbers here? We've got 2.9, 1.5. So we go 2.9, that was individual 1, and it is 1.5. And then we've got 3, 4, 5. So I'm going to show you in a minute and a half how to do a pie chart. Okay, 1.3, 1, and 1.2. 1.3, 1.3, 1, and 1.2. So first thing you do is you're going to add all of those. So we're going to zip this up a bit. So you add this up. So what have we got here? 9, it's 14. Hang on, I've got a calculator here. So let's quickly do this calculator. So 2.9 plus 1.5 plus 1.3 plus 1 plus 1.2 equals 7.9. So how do you do your calculations? Here you go. You're going to say 1 is going to be 2.9 divided by 7.9 times 360 is equal to. So 2.9 divided by 7.9 equals times 360 equals, and this gives you 132 degrees. Okay, and then I'm going to do another 1, and then I'm going to show you. So the 1.5 divided by 7.9. You're going to divide by 7.9 every time because 7.9 represents 360 degrees. So we want to know what portion of that each of these is going to make of the 360. So divide, so let's just quickly do this one. 1.5 divided by 7.9 is equal to times 360 equals. So that's going to be is equal to 68 degrees, etc., etc. You can do the same for each of those. Then you draw your pie chart with a protractor, and then you measure out 32 degrees, 68 degrees, and then you say individual 1, individual 2, and so you carry on. Remember your heading for your pie chart. There you go, folks. Have an awesome week. Be good. Stay safe. Stay warm. Take care. Mwah.